Good evening, everybody. Good evening, and welcome to tonight's event. Now, it's my job on behalf of the University of Southampton to extend a warm welcome to staff, students, alumni, supporters, and everybody joining us from around the world, and there are many. So my name is Jay Self, and I'm a professor of ophthalmology in Southampton, an associate professor, and I'm also a consultant ophthalmic surgeon. Now, I'm your host tonight, and I spend most of my time looking at children with eye diseases, both in research and in the clinic. Now, although most of my work is with children, we have a broad eye research group, and we look at conditions which affect people of any age, from the youngest to the oldest. Now, sometimes in clinic, when we sit at work at the hospital, we see patients with devastating eye disease, and this is quite common for us. And some of these eye diseases are even related to other health conditions elsewhere in the body. So as eye doctors, we spend our time trying to diagnose patients quickly and accurately, and then to give them sight-saving care. Now, we have two big problems with that. Number one is that for many conditions, we struggle to diagnose accurately and quickly. And number two, and perhaps most importantly, we don't have good treatments for a lot of the conditions we see, particularly in Southampton. So we're really pleased that you've all chosen to join us tonight and learn about what we're doing in Southampton to try and address those two problems of diagnosis and treatments. And we're going to hear about three very specific projects taken from a large portfolio of research that we do within our broader group. Now, one point is that it's really important to remember that all research starts with an idea. That idea then needs funding to prove that it's an idea worth following up. Then after that, this becomes a research project. And so everything we do goes through stages of needing funding, from the small to the large. And we're really lucky in Southampton that we've had the gift of site appeal for the last 20 years to help us with multiple stages along that pathway for almost every project we do. In fact, the three projects you're going to hear about tonight have all been funded by gift of site at some stage. So to tell you about those three exemplar projects, we've got a fantastic lineup of researchers this evening. One is Helena Lee, who will go first. Then we'll have Jorn Lekowski, and then Andrew Lotery is going to go at the end. Now, after these presentations, we're going to move to a Q&A session. That's a part of the event this evening. And so as the evening goes on, if you have questions, please do put them into the Q&A box, which you should see on the right-hand side of your screen. And we'll hope to get through as many of these questions as we can in the 25 minutes or so we have for Q&A at the end. So I think that's probably enough from me. I'd like to introduce our first speaker, Dr. Helena Lee. Now, Helena Lee is an associate professor at Southampton University and also a consultant ophthalmologist, and she specialises in neuro-ophthalmology, paediatrics, strabismus, and nystagmus. She has an international reputation for all the things that she does, and so I'm really excited to hear her talk. So over to you, Helena.
Good evening. I'm very excited to be able to share some of the work we have been doing at the University of Southampton to prevent blindness in children with albinism. Firstly, what is albinism? Albinism is a genetic condition that results in a lack of pigment in the skin, hair, and eyes. This lack of pigment also causes problems with retinal and optic nerve development. Consequently, the majority of people with albinism are registered as sight impaired in the UK. So what does retinal development look like in albinism? On this graph, we are demonstrating photoreceptor development over time. On the y-axis, you have thickness in microns. On the x-axis, you have age in months. In blue, you have the albinism group. In red, you have the control group. And what you can see here is that between birth and 18 months of age, there is a rapid development and elongation of the photoreceptor layer. In albinism, this lags behind the control group. However, the important point to note is that there is evidence of ongoing retinal development in albinism before 18 months of age. Potentially, if we can target this with the right therapeutics, we can normalize retinal development and prevent blindness in children with albinism. So the next question is, can we actually modulate or change postnatal visual development? And the answer is yes, we already do this when we treat lazy eyes or amblyopia. In lazy eyes, we are able to improve the vision in the weaker eye by using combination patching glasses. but this is only effective before 8 years of age. So what we need to work out is what our equivalent of patching is for albinism that can be given before the age of 18 months. So in order to do this, we need to understand a little bit of what is happening at the biochemical level with pigment synthesis. So this is the pigment synthesis pathway. And at the very beginning of the pathway, you have amino acid tyrosine, one of the basic building blocks of life. And this is converted by the enzyme tyrosinase to a very important chemical called DOPA. This is then converted again by tyrosinase to melanin, which is the pigment that you see in your skin, hair, and eyes. DOPA itself acts through the OA1 receptor to trigger a signaling cascade that modulates retinal development. And we see evidence for this in mouse models of albinism. On this graph here, we are demonstrating the levels of DOPA within the mouse eye. On the x-axis, you have postnatal age in days. In blue, you have pigmented mice. In pink, you have albino mice. And what you can see here is DOPA levels peak 1 month after birth in the mouse eye. This does not occur in the albino eye, as they are not able to synthesize DOPA like the pigmented mouse. And interestingly enough, this matches the therapeutic window that we have identified in our human children with albinism. So our equivalent of patching in albinism is levodopa. And what's nice about DOPA is that it's already in use as a drug to treat Parkinson's disease, as well as a number of pediatric neurological conditions. So our next step was to prove that giving levodopa can improve visual function in albinism if given at the right age. So in order to do this, we went back to our mice with albinism, and we gave them levodopa in their drinking water, and we tried this at 3 different ages, 2 of which falls within that therapeutic window, and 1 of which falls after. So this is at birth, postnatal day 15, and postnatal day 29. We checked their retinal structure and function using OCT and DRG examinations at 4, 5, 6 weeks of age, 2, 3, and 4 months of age. And what we got next was very exciting. So in order to understand what an ERG is, just think of it like an EEG, which measures the electrical function of the brain. The ERG measures the electrical function of the retina. And the higher the amplitude of the A and the B wave, the better the retinal function. So on this ERG, you're seeing the average ERG results for a group of control mice. In green, you're seeing the treated albino mice, and what you can see here is that there is a significant increase in the amplitudes of the A and B wave in the treated albino mice compared to the untreated control mice. And on the right, you have a bar graph showing the 3 different age groups, and this shows that in the treated groups at birth and 15 days of age, we are able to obtain significant increases in the amplitudes of the A and B waves, but not in the group that was treated after 28 days. So this was novel data confirming for the first time proof of concept for postnatal modulation of retinal function in albinism using oral levodopa treatment. Now it would be nice to also see this in action in terms of function. So in order to do this, we used a tool called the optometry, which is a way of testing visual function in mice while they are awake. Now it would be nice to get them to read the letters of a chart, but we just can't do that with mice. However, we can get them to follow stripes, and the finer the stripe that they can follow, the better their vision. So this is a video of a treated mouse, and if you watch her head carefully, you can see her following the stripes, and she follows them so enthusiastically that she nearly falls off the platform. Now, if this were an untreated albino mouse, they'd be very still. They wouldn't move at all because they actually wouldn't be able to see these stripes. So there she goes. Okay. And we subsequently went on to test 3 different human equivalent doses, as in doses that have previously been used to treat infants and young children with other neurological conditions. And on these graphs, you see these optometry results for 4 different directions, clockwise, counterclockwise, up and down. The frequency, which is basically how fine the stripes are on the y-axis, and the ages on the x-axis. The pigmented control mice are in grey, the untreated albino mice are in blank, and the different doses, the different human equivalent doses, are illustrated in different colours on these graphs: green, blue, and brown. And what you can see here is that each of the human equivalent doses, but in particular the medium and high dose human equivalent doses, improve the optometry results almost to the level of the normal pigmented control mice. So our conclusion so far is that treatment with human equivalent doses of L-DOPA, while there is still residual potential for retinal development, normalizes retinal development and optimizes vision. And we are now in the process of planning a pilot clinical trial using these human equivalent doses. Thank you, and I'd like to acknowledge the research group in Leicester as well as the research group in Southampton for all their help with this work.
Thank you, Helena. So you can see from Helena's presentation that a lot of the work that we do uses cutting-edge science. This is cutting-edge genetics, eye movement recording, all sorts of other cell biology to look at a condition that, in fact, the mankind has known about for thousands of years. And for the first time, we're getting insights into what's causing that condition and what we might do to help. And it's really fascinating, isn't it, to see that Helena's work has found that a drug that we already use for something else may well be useful for this condition as well. So that's repurposing. So fascinating work. Thank you, Helena. So the next talk we have is from Dr. Jorn Lukowski, and Jorn is a senior research fellow in vision science at the university. His principal interest is looking at developmental biology and genetics to try and find novel treatments for inherited retinal diseases. And some examples of those are a condition called retinitis pigmentosa and cone dystrophy. So without further ado, over to you, Jorn.
Hello, my name is Jorn Lukowski, and I'm a Senior Research Fellow in Vision Sciences. Although I'm a cell biologist by training, I have a long-standing interest in developing new therapies for retinal diseases. And towards that end, my group is using what's called stem cell-derived retinal organoids, which are essentially many retinas in a dish, like the one you can see in this image. And we're in particular interested in studying human development in order to come up with new therapeutic interventions. So for today's presentation, I'm going to talk about some of the work that's going on in my lab, specifically a project which revolves around the development of a drug-based therapy for retinitis pigmentosa using these stem cell-derived retinal organoids. So retinitis pigmentosa is a group of inherited retinal dystrophies and the leading cause of blindness. It is characterized by this progressive degeneration of the photoreceptors and pigment epithelial cells in the back of the eye. This leads initially to this characteristic tunnel vision, but can also progress to complete loss of vision. Unfortunately, there are currently no effective treatments available, and so there's an urgent clinical need to come up with new and better interventions. We have known about the disease progression for quite a while now. So in the healthy retina, the photoreceptors reside in the back of the eye, next to the retinal pigment epithelium. In this image, the rods that mediate vision in dim lighting conditions are shown in green, whereas the cones, which are important for high acuity and color vision, are shown in purple. In retinitis pigmentosa, mutations in rod express genes lead to the loss of rod photoreceptors over time, initially leaving the cones intact. But once all the rods have been lost, the cones start degenerating as well and die eventually. And so this then leads to the irreversible loss of sight. So, while we have known about the general cause of the disease, the underlying molecular mechanisms are much less understood. But we have learned from different animal models that rods produce and secrete a molecule called the rod-derived cone viability factor, which is important for the survival of the cone photoreceptors. This molecule binds to a receptor on the cell surface of the cones called basogen, and this in turn allows the cones to import glucose, sugars, into the cone cell, which is really important for their normal metabolism. As the rods die in the course of retinitis pigmentosa, this survival signal is lost, and the cone starts starving, and eventually it is lost as well. Now, this is our current hypothesis of this process. I think it's a very good hypothesis, but there are several issues. In general, the cone survival pathway remains poorly understood. Unfortunately, most of our evidence comes from animal models, and so the findings may not directly translate into the human context. But I think you can appreciate that understanding these mechanisms would be important and useful, and they may lead us to insights which would eventually allow us to replace the survival signal artificially in the form of a drug in order to save the cone photoreceptors. And so this is really the point of this project. So in order to study this process better in the lab, we're using what's called retinal organoids. And those structures are grown from pluripotent stem cells. There are many different types of stem cells, but pluripotent stem cells are very special because of what they can turn into. They can be derived either from a recently fertilized egg or through reprogramming of somatic cells, typically skin cells, from a human patient. And either case, they have the ability to self-renew pretty much indefinitely, but also give rise to all the different cell types you find in the normal human body. And so basic research has really provided us with a great deal of knowledge and also tools to generate quite complex different tissue types and in some cases parts of organs. And so these organoids are really human retinas in a dish, because they contain all the main cell types you also find in the human retina in the correct spatial organization. And they now provide us with a necessary model system to study both normal human development, but also diseases, and importantly help us develop treatments for a range of different retinal diseases in a way that hasn't been possible before. So a real game changer. So how can we mimic retinitis pigmentosa in our organoids? Well, to achieve this, we took advantage of this genome engineering tool called CRISPR-Cas9. And you can imagine CRISPR as these small molecular scissors that allow us to cut the genome in very specific ways. And for this project, we decided to cut out the gene that encodes for the receptor for the survival signal, called basogen. So we hypothesized that genetic removal of the cone survival signal receptor would result in retinitis pigmentosa-like conditions in the organoids. And this in turn would enable the search for chemical compounds or drugs that would enhance cone survival. In a nutshell, this approach was successful. As you can see here on the left-hand side in this gel analysis, we have found that in our mutant organoids, the gene that encodes for the survival signal receptor is no longer active, and analysis of our organoids using antibodies for photoreceptors also shows that the genetic removal of this receptor results in reduced survival of photoreceptors. And so, taken together, this indicates that our model system is working as designed. In addition to the mutation, we also introduced a fluorescent reporter gene in our cone photoreceptors. For that, we essentially molecularly tagged a gene which we knew was expressed only in photoreceptors, which is called GNGD2. And so wherever we see GNGD2 present, we will now also see this red fluorescent signal. And this is very useful because It enables us to track and visualize in real time, as you can see here, our cone photoreceptors in the organoids. And importantly, it also lets us isolate these cells now through a process called flow cytometry. So we can now obtain very pure populations of cone photoreceptors, which is very important for the downstream applications. So we are now in a very good position to carry out a drug screen. We will essentially grow many organoids and generate lots and lots of cone photoreceptors, which we will then purify using flow cytometry. These purified cone populations will be seeded in these multi-well tissue culture plates. We will then starve the cone photoreceptors, just like what happens in retinitis pigmentosa, and at the same time treat them with a chemical library, which is comprised of about 2,000 chemical compounds, which are already clinically approved. And then we want to identify the few drugs which will actually preserve cone viability and enhance survival. And those drugs will then be further optimized and will hopefully form the basis of a future drug therapy for retinitis pigmentosa. Now, I should mention that all this work is currently being conducted in our stem cell laboratory, which was generously funded and supported by a gift of sight. So we're very grateful for this. It allows us to operate at a very high level of quality, but also scale, which is really important for these type of projects. Now I'm coming to the end of my presentation. I'd like to thank you for your support and also acknowledge some of the people who have been involved in this project, both here at the University of Southampton and beyond. Thank you very much.
Thank you, Jorn. Amazingly colourful pictures we get from the laboratory at times. I'm sure you'll all agree looking at those slides. Now, the other interesting thing I think from that talk, fascinating work, is that we have the technology now to turn cells into other cells. And that's a real bonus for research and what we try and do. And also, as Jorn mentioned there, this use of retinal organoids, which is a fancy term for basically making a little eye in a dish, is incredibly useful. Because if you think about it, when we're testing treatments, we can test them on a cell, but when you test them on a patient is much more complicated. So these models used to develop and test drugs that are much more like the tissue we're interested in are invaluable, and that's a really good example of it. So thank you, Jorn. Now, our final talk this evening is from Andrew Lotary, who most of you may know. He heads up the Vision Research team and is Professor of Ophthalmology at Southampton. Now, Andrew has been a mentor of mine. I've been working with him for about 20 years, and although we do different work in terms of the age groups that we look at, Andrew's been a fantastic mentor to me, so it's a privilege to introduce him. He's going to talk about some of his latest research, looking at a common disease and a group of common diseases, which we've been talking about rare diseases so far tonight, and he's going to talk about some of the cutting-edge research and also clinical trials. So, Andrew, we look forward to hearing your update. Over to you.
Well, thank you for joining this Beating Blindness Lecture 2022 from bench to bedside. And this is really a special lecture because it actually reflects 20 years of eye research at Southampton. And so I've just put up a slide of one of our first research projects 20 years ago in Guernsey, where we brought a lot of equipment over to the island to take blood samples and examine patients with macular degeneration. And that's because we believed that understanding the genetics of this disease would help us to develop new treatments. And so we would go to the island and then come back on the ferry, and this is us to the amusement of other ferry customers, sorting blood samples at about 1 o'clock in the morning, going to Weymouth. And then we'd take those samples back to the lab where they'd be extracted to DNA, and this was done by Helen, one of my research technicians, and then we'd perform experiments on them. And I just want to mention how valuable gift of sight was to this whole process. So while we had a grant to do this work, these grants obviously come to an end and new grants have to be applied for. And it would have been impossible to keep staff employed without bridging funding from the Gift Aside appeal. So Helen, shown here, has been with me for 20 years, and that's only been possible because of Gift Aside. And she has made amazing contributions during that time to our work, extracting thousands of DNA samples and doing many other projects as well. And without Gift Aside support, that would not have been possible. So what did we know back in 2002 about this condition, age-related macular degeneration? Well, not very much, I'm afraid to say. We knew it ran in families. There was often a family history if you asked. Unfortunately, people went blind with the condition. There were no treatments, and it was the commonest cause of blindness in the elderly in the developed world. And that's really all we knew back in 20 years ago. So it's quite a tough condition for patients to have and for doctors to look after. But what do we now know about age-related macular degeneration after 20 years of research? We know that it's caused by over 52 different genetic changes, and that gives us an insight into the cause of the disease and ways to treat it. We can use new techniques which didn't exist 20 years ago, such as artificial intelligence, to understand AMD. And blind registrations for the first time in history are actually decreasing because of the progress being made in terms of treatment. And there are things you can do to reduce your risk of progression if you are diagnosed by AMD, even if you have a genetic risk factor. And that includes not smoking, exercise, eating a Mediterranean diet, taking certain vitamins such as VitIce2 or Ocuvite Preservision. And we know that treatments for wet macular degeneration work if they're started early. And we know that treatments for dry age-related macular degeneration are on the way. And that's all because of the research that has gone into developing these treatments. So this is a photograph that a patient gave me a few weeks ago, Peter. And Peter, unfortunately, has this large spot blocking out his central vision. And as you can see, he's a talented artist, and he also likes working with his hands. But everywhere he looks in the center of his vision, there is this hole in his vision. So why does Peter have this hole in his vision? Well, we know now that if you look at the structure of the retina, there are blood vessels beneath the structure that senses light, the retina. And these blood vessels, under the stimulation of a growth hormone, come forward like weeds coming through cracks in the pavement, and they come into the substance of the tissue that senses light and leak fluid and blood and eventually form a scar, which causes this central hole in your vision. Now, happily, we have a treatment for this, and that is to inject a drug to inhibit this growth factor, and we do this under lots of anesthetic. But unfortunately, until recently, we've had to give that drug every month or every couple of months to stop these leaky blood vessels coming back. And if you start this treatment early, you can get really good results in terms of keeping people's vision and improving their vision. But it does require a bit of a marathon in terms of treatment with lots of injections. So we've been doing research along with colleagues all over the world, and this is our clinical research team. And this is me holding a certificate that the hospital gave us for being one of the best research teams in the hospital. And part of this research has recently found that a new drug treatment called faricimab can extend interval between injections so that about 80% of patients now only need an injection every 12 or 16 weeks, which is a huge step forward if you have to have these injections. We're also looking for new treatments that might even last longer, such as gene therapy. And I'm just showing some slides here showing how with gene therapy we can inhibit blood growth in the lab. And this would be a one treatment, and then you're done treatment for wet macular degeneration. And this is just starting to go into clinical trials as well. And then we are performing clinical trials of new treatments for dry age-related macular degeneration. And these aren't available yet in clinical use, but I'm hopeful in the next few years they will be. And to do this research, we need a very specialized camera shown here. And this camera was paid for by the gift of sight appeal. And this costs about £100,000, so it's not a cheap piece of equipment. But it helped us secure a grant of £4 million from the Wellcome Trust to do some of this work for dry macular degeneration. And so that's a 40-fold return on the investment. So I think it's a pretty good deal. And so we're also trying to understand AMD better using computer science, and that's because of the volumes of information we're dealing with now. This one retinal scan I'm showing here contains 1 million pieces of information, and we're looking at over 500 million of these scans. And the only way to do that is with computers, so we're working with computer scientists all over the world actually to identify early changes in these scans that would help us better manage patients with macular degeneration. And because of all this work that we're doing in Southampton, external organizations have assessed our contribution, and so they look at the papers that I've published, which of course is the papers that my team has published, so not just me, and it ranks us in the top 0.079% of authors working on macular degeneration around the world. So Southampton is really world leading in this research. So the future for Peter and for patients with macular degeneration is that wet AMD treatments are now extendable out to 16 weeks between injections, and we expect that in the next year or so that will go out to 6 months or even a year between treatments. Dry AMD treatments that stop dry AMD progressing are in clinical trials, and I think show a lot of promise. And this is based on our genetic research, going way back to those early days in Guernsey, where we identified complement gene changes in those patients from Guernsey. And earlier diagnosis and treatment using genetics and computer analysis of retinal images, I think, will be part of our management of patients in the future. And so this will all lead, most importantly, to better outcomes for patients, and blind registrations will come down further. So to summarize, we're using a wide variety of techniques to develop better treatments for a range of eye conditions. And I've only been able to talk about macular degeneration in this presentation. We studied disease at a very basic level in the lab with stem cells and genetics. And we use clinical data and the latest artificial intelligence science to study unbelievable amounts of information. And we do clinical trials to confirm results so patients get the right treatment. And this is our from bench to bedside approach that we've had over the last 20 years. And gift to sight is essential to all of this and helps us to do this work as quickly and as productively as possible. Thank you very much for listening.
Well, thank you, Andrew. That's an amazing talk. Again, it's mind-boggling, isn't it, to look at the number of images that we need AI and machine learning to look at to do this science now. And I think it's a really good example of how we try to stay abreast of the latest technology and tools which are available rather than doing the same thing we've always done as researchers. So it's absolutely fantastic work, really interesting. Thank you. So we are drawing towards the question and answer section of the evening now. So I would encourage people, please, to put questions into the question box. We've had quite a few come in, but please do put more in. And we're approaching that time where we'll start distributing these questions. And I'm going to pick on our poor lecturers this evening and see who wants to answer what. So I think we'll start that process, but please do keep bringing those questions in as time goes on. So the first question I have at the top here, team, is does your research team collaborate with other scientists working on vision loss around the world? Andrew, maybe you'd be good to answer that one.
Absolutely. It always has done. And for the grant I mentioned from the Wellcome Trust, for example, has senior investigators from the United States, from Vienna in Austria, in Switzerland, as well as many sites around the UK. We have collaborations with scientists in India. I was on a Teams call with researchers in India this morning. We collaborate, and I think I speak for everybody here, with scientists all over the world because research is a team sport. It is definitely a team sport, and we get results quicker if we work with people. So we are very collaborative.
Fantastic. Thank you. That's a really good answer. It'd be really interesting one day, wouldn't it, to draw a map and put a pin in it for every country where we collaborate with people. And it is absolutely bewildering, isn't it, the number of places that we have connections with through research and through the clinics as well. Thank you. So the next question I have here is, how important is it for the scientists to be located at the hospital close to patients? Jorn, I wonder if you might want to have a stab at that one.
Well, yes. So obviously there's a great advantage to be very close to patients, but also to the medical personnel. So like yourself or Professor Lodry, it's always good to have the clinician's point of view for our research. That's really critical, and many research projects are actually done in collaboration. So my research project in particular is very closely associated with Professor Lodry, and really kind of was spawned from a conversation really between the two of us.
Fantastic. Thank you. Did you have anything to add to that, Helena or Andrew?
I think it's really important because the patients are constantly feeding into our research. So it's so useful to be on the same site doing our research as where we are also seeing and treating our patients.
Yeah, and I think we are somewhat unique in having both basic scientists and clinicians co-located on a hospital site, and that does really help to bind the team together, and also, you know, for exchange of information. So I think it's really important. And as I mentioned, patients have given me literally thousands of blood samples for DNA and research and genetics research, and that would have been much harder to do if the eye department was not co-located in the same area as the laboratories for processing all those blood samples. So I think it's a real advantage.
Absolutely. Thank you. So the next question we have is, how much funding does vision research get compared to other diseases? Now, I'm sure we've all got our thoughts on that, but I suspect they're very aligned. Andrew, do you want to have a go at that one?
Absolutely. Well, ophthalmology performs about 10% of all NHS outpatient activity and 10% of all NHS surgery, but we get less than 1% of national funding for eye research. So compared to the contribution that ophthalmology makes to the NHS, we get about 1/10 of the funding. So I think it's well recognised that ophthalmology gets relatively little funding, and the economic benefit of funding research has been looked at by experts, by external reviewers, and there's a huge benefit in preventing blindness in terms of not just because it's the right thing to do, but the financial benefit is huge too if you can prevent people going blind. So there is a disconnect between the amount of funding we receive and the need and the benefits that can come from it. So that's where Gifticide is incredibly helpful for us to support our work and get it going.
Yeah, absolutely, and I'd agree with that. And I think the other thing that's interesting is that COVID has actually widened that gap slightly, isn't there? There's reasonable evidence that actually COVID has made that disconnect even greater for various obvious reasons. So absolutely, prioritising funding into eye research is something we all try and really push because it is a big problem. Thank you. So the next question we have is, are there any developments that would address sight loss due to optic nerve damage? I think you know where I'm going with this one, Helena. Would you mind answering that for us?
Yeah, that's a tricky one because optic nerve damage comes from a whole variety of diseases or trauma. It could be glaucoma, could be blood flow problems, it could be toxic problems, it could be genetic problems. So it really depends on our understanding of where it's come from, where we can address those causes. And then I think what people are looking at is the possibility of regenerating optic nerve, but we are a very long way away from that. I think Jorn's work is probably the closest to that, and that's kind of working on stem cells, but even then trying to get something to regenerate is quite a tricky process. But as always, it's understanding where the problem has come from in the first place, whether it was genetic, whether it was blood flow, a trauma.
Yeah, absolutely. Thank you. Sorry, Andrew, did you want to say something on that?
Yeah. Well, just we're talking about collaborating with people. I was at a meeting. I was invited to speak out of Jerusalem last week, and I heard about recent clinical trials on a genetic condition that affects the optic nerve, which is showing some promise for a condition called Leber's hereditary optic neuropathy. So there is some progress, but as Helena says, it's slow. It's challenging, but it is exciting. I mean, I did hear of clinical trials that are showing some benefits for patients with one specific form of optic nerve disease. So that's great.
Thank you. And just to add to that, I think the thing to think about with optic nerves is that there's three ways of addressing it. Number one is thinking about reducing further loss of nerves, which is one sort of stream of treatment. One is about reconnecting or growing new nerves, which is traditionally an extremely complicated and hard thing to do and connect. And the third thing is that depending on age, if your optic nerve hasn't developed by the age of 8, if you did have a perfectly functioning optic nerve after that point, you would still not see through that eye because the brain has never learnt to see. So there's a whole world of complexity built into that, isn't it? But yeah, thank you. That's a really good question. So the next question I have, again, this might be heading your way, Helena, is there a correlation between TIAs, that's transient ischemic attacks, mini strokes, and eye conditions?
Okay, I would say yes, there's multiple eye conditions. If you think about a TIA, it's basically a problem with blood flow. And the eye is, like the rest of the brain and the body, very dependent on having normal blood flow and normal blood pressure. So if you're someone that suffers from TIAs, it could also mean your eye could be vulnerable to conditions that affect blood flow. So things like things that affect the optic nerve. It may mean that you have diabetes because that can affect your blood flow. It could mean you have high cholesterol, and that can cause blockages in some of the blood vessels in the retina. And we keep going on, but this basically, the eye, like everything else, also is reliant on blood flow. So yes, short answer.
Fantastic. That's great. Thank you. So these questions are jiggling around. So I'm just trying to find the next question. Let's see here. Okay. So are there any, sorry, we've answered that one. Is there any connection between sight loss and cognitive function? In other words, memory loss or changes given how much we use our vision to store and draw on memories and information? Shall we give Helena a break? Do you want to have a go at that one, Andrew, and we can have a think?
I can have a start. Just because I can see from 2 points of view. One, the cortical side, and the second, the retinal side. So maybe if Andrew does retinal and I can do cortical, because basically I do see people with Alzheimer's present to the neuropathic clinic. And that's because Alzheimer's can actually affect the visual areas of the brain that allows you to understand and process what you're seeing. So definitely link at the cortical point of view. And there is a link with the retina. So I'll pass that over to Andrew.
Thank you. Well, there is, with new ways to image the eye, we do see changes in the blood, defined blood vessels in the back of the eye that are correlated with having Alzheimer's disease. So some of the earliest changes can actually be picked up in the eye, which is really interesting. We know that certainly hearing loss is a risk factor for developing Alzheimer's disease, and I don't think the research is so well defined for sight loss, but I think it's quite likely that if you've got reduced sensory input from eyes, just like hearing, it may be a risk factor for Alzheimer's. I'm sorry, there's someone letting off fireworks outside. I hope you can't hear that, but okay, good.
That's great. Thank you. Thank you for those answers. So next question is, considering the amazing progress you have all made over the past 20 years, where do you expect eye research to head next? That's the big question, isn't it? Perhaps we could answer those separately for our individual fields. Do you want to have a go at that one broadly for your field of interest, Jorn? And then we'll move around.
Yeah, I think it's a really good question. I think it will. So the stem cell field will definitely expand, growing retinal organoids, producing essentially retinal tissue for all kinds of applications, and also therapeutic applications later on. It will definitely expand. There will also be potentially genome engineering to fix genetic conditions. So I think there's a lot of exciting and very promising research going on, which will hopefully soon be translated into the clinical environment as well. It's a very, very active area.
Thank you. Helena, do you want to have a go at that one? Anything to add?
Yeah, so what I think will happen in my area, and I span peds and neuro-ophthalmology, I think we're going to make diagnostic advances. So we'll understand exactly where everyone's sight losses come from, whether it's genetic or otherwise. Then I imagine I'll go to someone like Jorn and go, can you make me a model of this person's disease? And then I'll go, right, okay, let's test this range of different therapies that we have, whether it's gene therapy, cell therapy, or nanotechnology, whatever, on this model in a dish, and be able to actually personalize the medicine that we give and be able to prevent vision loss in children and perhaps help restore vision loss in older adults.
Thank you. Andrew, anything to add?
Yes, I mean, I agree with everything that's been said. I think we'll be able to diagnose disease earlier and treat earlier. At the moment, I sometimes think our therapies were a little bit like the fireman who comes along after the house has started to burn down, and we're really intervening at a very late stage in eye disease. And I think we're going to be able to intervene earlier and prevent people getting to these more sight-threatening stages of disease. And that's going to come from better genetic diagnosis, better imaging of eyes, and better detection of early signs of disease. And then, I'm very excited about gene therapies. We have one licensed on the NHS now for one very specific form of eye disease. I think we're going to see a lot more of that in the future. And I think also eventually we will develop stem cell therapies thanks to people like Jorn and their research and understanding how to make these cells integrate into the eye. And then, as I mentioned, computer science, we're going to have much better automated diagnosis as well, which will help doctors to manage with all the patients. And we're going to have treatments for some conditions we can't treat very well yet. I mentioned dry age-related macular degeneration, for example. And I think that's going to be the next big area of treatment in my field. Thanks.
Thank you. Yeah, so some great answers there, and a lot of what we're describing there, I guess, is what we would call personalised medicine or stratified medicine, where we're identifying which patients would benefit from which thing at the right time, and ideally before it becomes a problem. So that does seem to be a general theme for the way that medicine is going forward, isn't it? Fantastic, thank you. So we have a couple of questions here that are related. Perhaps there's one about the link between high cholesterol and the use of statins for AMD risk, and one question asking about the use of vitamins in AMD. They're close to what we've just been talking about, aren't they, in some ways, Andrew? Maybe you want to have a stab at those?
Yes, well, interestingly, some of the first research in statins and AMD was done in Southampton by one of our colleagues, Nigel Hall, more than 25, 30 years ago. And so people who are on statin drugs tend to have a lower risk of having age-related macular degeneration. And it seems to be there's an anti-inflammatory effect of statins. So taking statins is certainly good if you've got a high cholesterol, but there may also be benefits in terms of reducing inflammation as well, which helps reduce the risk of age-related macular degeneration. In terms of vitamins, there were 2 large-scale studies performed in America, the National Eye Institute, which showed that combinations of vitamins, vitamin C and E, mixed with copper and zinc, lutein and zeaxanthin, slowed progression of age-related macular degeneration. So for patients who have moderate signs of AMD, they seem to help to reduce progression to the late stages. So there are several companies that make the same vitamins that were used in these clinical trials. One's VitEyes 2, another one's Ocuvite Preservision. Unfortunately, they're not available on the NHS, but it's something that perhaps patients might want to consider taking. You can also reduce your risk as well with a diet that has leafy green vegetables, particularly not smoking. That's a much higher risk factor. So all these things can help.
Thank you, Andrew. So the next question I've got here is anyone researching visual deficiencies caused by anti-inflammatory substances? Is it a recognized phenomenon? Does anybody know or like to offer anything about that? Anti-inflammatory substances being a cause for visual deficiencies.
Well, people have looked at aspirin, and there's been conflicting evidence whether it is a risk factor for macular degeneration or protective. I think really we haven't really, the jury's out. We don't really think it increases your risk of macular degeneration, but it does not help either. I'm not aware of other research about anti-inflammatory drugs being detrimental to vision. I don't know if anybody else is, but no.
But there is work, isn't there, looking at use of anti-inflammatories as a treatment, in fact, for
Yeah.
a number of eye conditions.
Absolutely.
Some not. Okay, thank you. So the next question I have here, I'm sorry if this order is not correct, but I'm doing my best with the ordering here. Both my parents had macular degeneration. What are my chances of getting it?
So it's a complicated disease. There are many genetic risk factors. So if you have a family history of macular degeneration, you are probably at a slightly increased risk of developing it yourself. But it really depends on what genes you inherit. Some are much more likely to increase your risk than others. Unfortunately, we can't do genetic testing as yet for macular degeneration because it's so complicated with so many genes. But as I said, we do now know that even if you have a genetic risk factor, if you make lifestyle choices, you can still reduce your risk of developing the disease or progressing by eating a healthy lifestyle, exercise, not smoking, Mediterranean diet. These all can reduce your risk even with a family history, and that's something maybe even more to think about. And then other than that, I think it's just regular eye checks with an optometrist over the age of 50. So if very early signs are picked up, you can be referred to hospital for assessment of potential treatments.
Thank you, Andrew. Yes, so I guess to summarise that, just to chip in my bit on that, some medical conditions that you hear about are inherited to some degree, but it's complicated, and macular degeneration is one of them. Some medical conditions are inherited in very clear ways. This is where you hear about 50% of children, or 1 in 4 children might inherit it. So AMD is an example of a complex disease where there certainly is genetic contribution, but it's not that clean or clear in families to give a specific risk. So thank you. That's a really good answer. Thank you, Andrew. I have a question here that may be relevant for you, Helena. Is there any further understanding of Charles Bonnet syndrome? It might be worth just explaining what Charles Bonnet is before you answer that, Helena.
Okay, so that's basically I think actually Andrew probably sees more of it, but because we primarily see this in patients with macular degeneration, and it's almost like your brain is showing you images that you want to see, although you can't see, to try and simplify it. And it was the question was, do we understand more about what's happening?
Yeah.
Right. So I'm actually hand that over to Andrew.
Excellent.
Yeah, thank you. I mean, it's really interesting. Patients with severe sight loss from macular degeneration often don't volunteer that they have visual hallucinations until you ask them, but it's thought that up to 50% of patients with severe sight loss from macular degeneration have this problem of visual hallucinations, which can be amusing to some patients or quite distressing. And I think it's important always to ask patients, do they have visual hallucinations? And they're very relieved to understand that they're, it's not a psychiatric condition. They're not going mad. I think it's the brain trying to make sense of a garbled picture, like a slightly mistuned TV, and generating images from the limited information that comes in from an eye that has severe macular degeneration. There is ongoing research into trying to develop better treatments for it. There isn't really, I would say, a good treatment at the moment, except just to tell people, explain what's happening, and that can be reassuring. Sometimes it reduces with time, and people get used to it, or it doesn't bother them so much with time. A few patients have tried antidepressants as a potential treatment. It seems to help in a small number of patients, but I think more research is really needed to know how effective that is. But it's important to discuss it with patients so they know that it's part of their macular degeneration problem and not a separate psychiatric problem.
Great, thank you. I think we have just time perhaps for one, maybe two questions left. One is, are there ways of promoting visual health around the world and the UK? And that's got a few thumbs up, so a few people interested in that one.
I mean, there is an annual there are annual days promoting sight loss or trying to stop it, that are national programs to draw awareness to the challenges of having sight loss. There is work. I mean, there's Jay, among others, have gone out to do cataract camps in developing world. Royal College of Ophthalmologists here in the UK has strong connections with African colleges of ophthalmology and tries to promote education and research there. So yes, there are definitely efforts to highlight the challenges of sight loss and to give some of our expertise to doctors in the developing world as well.
Absolutely, thank you, Andrew. And I think there really is a priority nowadays, isn't there, into teaching a man to fish rather than giving him a fish. So a lot of the work in developing countries is about providing infrastructure or training, setting up local resources to make things sustainable. But yeah, absolutely, thank you. So I think we're just drawing to the end now. So anyone who's not had their question answered, I'm very sorry about that, but please do send them in. We can get back to you via other routes. That's what we've done in previous years. The final question, which has come in very fortuitously from Anne Moran, which is, how can we contribute to Gift of Sight? And I think that's a fantastic final question. Thank you. So the answer to that is multiple. The most important way is to stay in contact with your inbox, because there'll be follow-up after this lecture. Keep coming to our lectures, stay in contact with Gift of Sight. So from me, I'd like to say thank you to everyone for coming. Andrew's going to give final thoughts in just a second. But from me, thank you to all our speakers. You've been fantastic, and thanks for letting me put you on the spot. Thank you for all our fabulous questions. And just to remind everyone, there's a poll at the end which will come on your screen. Please do fill that in, because it helps us make this better every year. So thank you, and over to you, Andrew.
Thank you, Jay. As ever, I'd like to thank the supporters of the Gift of Sight Appeal for their help in funding our research. Your support is absolutely vital to the work we do. I hope tonight's lectures have given you a flavour of our research from bench to bedside and from young to old, and the goal it has of beating blindness. So thank you again, and I look forward to meeting you at our next event. Until then, good night.